Good afternoon, everybody. So the plan for today is to start a new taxon, the animals. We'll be spending several days on the animals. But first, I wanted to follow up on just a couple of last points about the fungi. So I ran out of time in just the last few seconds. We were discussing this particular life history cycle. So this is figure 32-12 from your textbook and is a representation of many of the events that occur during a fungal life cycle, although not all of these occur in every fungal life cycle, and there are variations, and I hope that you do take time to walk through some of the other life cycles. That'll make much more sense to you when you compare this particular more generic life cycle with some of the more specific ones. You'll see variations on the theme. I did want to highlight a couple of things that were peculiar to the fungi. I got as far as discussing these two, plasmogamy, when mycelia from two different individuals, two different mating types within the same species of fungus come in contact with each other. They can fuse in such a way that the cells that are a result of continued cell division after fusion of the mycelia end up with haploid nuclei from both of the original individuals prior to fusion, and that this is referred to as N plus N because the haploid nuclei are still distinct, and then the name given for this is heterokaryotic. So you should certainly know those names, plasmogamy and heterokaryotic. The last part that I did not have time to go for was this phenomenon here, karyogamy, which is the eventual fusion of nuclei which can occur at some point later. So in other words, this heterokaryotic condition can last for some time. It's not necessarily short-lived. The only other thing I wanted to show you in this particular feature, and of course as you're looking at a structure like this, you should be thinking to yourself, well, the mycelium are often buried in the surface, whatever it is, it's a food source. So for example, it can be underground. But there are spore-producing structures during both asexual and sexual reproductions. Both the asexual reproduction cycle shown on the left and also the sexual reproduction cycle shown on the right, both of which generate spores, and this is the stage that is dispersed, right? So it can be dispersed, for example, in the wind. The wind can blow. For dispersal to take place, of course, these spore-producing structures must be aware. If it's an airborne dispersal as opposed to aquatic, then these reproductive structures then have to be sticking out into the air. And so one can end up with a mycelium that is underground, and then the reproductive structures come above ground, or any kind of structure that's releasing a dispersal unit, such as the spores, will be above ground. So to make more sense of this, I thought I'd show you this picture, which is not from your textbook. It is a picture of a phenomenon referred to as fairy rings, and what this is when one ends up with a circle of reproductive structures of some fungus, such as this picture right here. And now that you have learned about the basic body form of a fungus, probably many of you are understanding how such a structure can occur. So if, for example, a spore had landed here in the center at some time earlier, fungal spore, and then below ground the mycelium grew and grew and grew, and it grew in all directions, then if conditions changed so that this particular type of fungus was triggered to generate a reproductive structure above ground, then this could occur at the periphery of the mycelium as shown here. So one can get a structure like this. So the reproductive structures can be sticking out of the ground or the food source, and the mycelium can be not visible from the outside as shown here. Okay, let's go ahead and do a clicker question then. I would describe this as very straightforward. Again, another example of a former test question, and this is the kind of thing one should be able to answer about a fungal life cycle, given what you've learned about fungal life cycles. Okay, consider this, that spores are produced for both asexual and sexual reproduction of fungi, Which of the following is the most accurate statement about genetic variability in the spores? I've given you 3 different choices here, and I've underlined the main differences between these 3 possible answers. A, spores are likely to be of comparable, meaning similar genetic variability, whether generated during asexual or sexual reproduction. B, spores produced during asexual reproduction are more likely to be genetically variable. Or C, spores produced during sexual reproduction are more likely to be genetically variable. So which of these 3 do you think is the most likely? If you're not sure, good time to chat with your neighbor, compare answers. I'll give you 15 seconds or so to chat about it and click in, and then I'll see what you guys think. Okay, everybody should be clicked in now. I'm hoping for 100% right here. Let's see. Fantastic. Congratulations, yes. Good job, everybody. Now, of course, you have had a very similar kind of question in a different taxonomic context. And so hopefully this is then reinforcing the concepts you've been learning about earlier, that one of the hallmarks of sexual reproduction is in fact generation of variable offspring. So nice job, everybody. You clearly have that concept. And now, the animals, a new taxonomic group for us. The plan for today is to start with the common features of animals. We'll consider the evolution of animals from a protist ancestor, and we'll consider what is meant by a body plan. So in order to understand body plan, we'll spend a little time talking about animal development, very early development primarily, and we'll consider some of these phenomena that take place during embryonic development, gastrulation, coelom formation. We'll also consider symmetry and cephalization, and the sponges will be the first phylum or major body plan of organisms of the animals that we'll go through. So first let's consider what is an animal. How can you recognize an animal? You can recognize an animal because it's heterotrophic, right? It's getting organic nutrients from other organisms or from the environment. It's not simply synthesizing its own organic molecules. Multicellular and eukaryotic. So far we haven't differentiated animals from fungi, right? Fungi are also heterotrophic, multicellular, and eukaryotes. But here's the rest of the definition of animals that ingest food and also lack cell walls. Now, who has cell walls? Well, plants have cell walls, and you have learned about some of the important chemical compounds that are in plant cell walls. Plant cell walls contain cellulose and lignin, right? Remember that? And fungi also have cell walls. And the cell walls in fungi do not contain cellulose and lignin, but they contain chitin, which is another important molecule that we have spent a little time talking about. Remember, cell walls are structures outside the plasma membrane, the cell membrane. Animals do not have cell walls. There are over 1 million different species of animals that have been described and named. This is thought to be only a fraction of the total number of animals that are on the planet. It is thought that at the current time there's on the order of maybe 10, 12 or so million different species of animals. Of course, you might think, well, how on earth can you get a number for this if they haven't been named yet? How can you count them? These kinds of estimates are based on the rate at which new species can be described if, in fact, appropriate taxonomic specialists go into a new area and describe species and then extrapolate based on how many new species they're finding per area or per unit time. Of course, this is just a guess. You can see guesses that are much larger and much smaller than this. So how are animals then related to the other living groups? This is figure 33-1 from your textbook in front of you right now, which shows, hopefully by now, the extremely familiar phylogeny of many of the eukaryotic groups. And again, fungi and animals are very closely related to each other, but not as closely related as animals are to another group called the choanoflagellates. And this is a group of protists. So this is the sister group for the animals. And therefore, to understand the early evolution of the animals, it's helpful to learn a little bit about this interesting protistan group, the choanoflagellates. Okay. This is figure 33-2 in front of you right now from your textbook, except that I have taken most of the branches that occur in this part of the diagram, and I've collapsed them all into just a single branch so it's not quite as distracting. So we have all of the animals here in this monophyletic group here, and then the sister group to the animals, the choanoflagellates, shown up here as the branch at the top. Now, it has been recognized for quite some time that choanoflagellates are probably closely related to at least one particular kind of animals, and that is the sponges, because of extraordinary similarity between the choanoflagellates and some of the cells of the sponges, and that's shown diagrammatically right here. So what you're looking at in the top of this figure is a colonial choanoflagellate. How do we know it's colonial? Because there are a number of different individual cells that are making this up. This is one cell right here. Choanoflagellates either occur as single cells or as colonies. They can be attached, as shown here, or they can be free-floating in the plankton. But regardless of whether it's a solitary individual or a member of a colony, they all look like this, where the cell has these little cytoplasmic extensions right here that form this structure, which is referred to as a collar, and inside the collar is a single flagellum, as shown here. And if there is a colonial choanoflagellate, then all of the individual cells that make up this colony are exactly the same. And that is how one tells a colony from a multicellular organism, right? So here's all the animals here. And notice that multicellularity is a synapomorphy that is a common feature for all of the different animals, including the sponges. How can you tell something is multicellular versus colonial? As you may recall, something is multicellular, in addition to just simply having more than one cell, the cells have to be different from each other. So if you look at the wall of a sponge, which is shown here, this is the body form of a sponge with a big cavity in the middle here. So this is the wall. If you look at the individual cellular components of the wall, there are different cell types. There aren't a lot of different cell types. It's a relatively simple body form, but there are in fact different cell types, unlike in a choanoflagellate. And some of the cell types are so-called collar cells or choanocytes, and they are virtually identical to the choanoflagellates. Look at this as shown here. There are cytoplasmic extensions that form this structure like a collar, and then there is a flagellum in the middle of this collar as shown here. So this similarity then, again, is one of the reasons why choanoflagellates have been thought to be closely related to animals for some time. Now, before I leave this figure, I want to point out that the sponges are the most basal groups of the animals as a whole, meaning that they are less closely related to the rest of the animals than the rest of the animals are to each other. Choanoflagellates have been recently featured in the science section of the New York Times, being highlighted because, in fact, it is recognized because they are a sister group to the animals that there are clues then to the early evolution of the animals in studying choanoflagellates. And I like the pictures here, so I thought I'd show them to you. Here's a picture of a, as opposed to a diagrammatic representation, here's an actual picture of a choanoflagellate. This is a colonial one. You can tell because there's a number of different choanoflagellates. If we look at that in higher magnification, you can see them here. Each one of those is an individual choanoflagellate cell. There's the flagellum. And then over here is an example of a solitary choanoflagellate. Over here on the right, I have a little animation running so you can see the flagellum beating. And so how do these choanoflagellates actually make a living? Before I go into that, actually, let me show you the last sentence on this figure legend here, so that you understand that choanoflagellates are not rare, obscure animals, protists. The last sentence here is there could be millions of choanoflagellates in a gallon of seawater. You might think, well, then why have I never noticed these? Because they're tiny, it's a single cell, they're microscopic, you wouldn't have noticed them. But if we consider now how choanoflagellates make a living, these protists make a living by filtering organic particles from the surrounding water. And how do they do this? Well, their flagellum beats like this, and as it beats, it draws water through this collar. This collar is actually a filtering device. So the little tiny particles, food items such as individual bacteria or small little bits of organic detritus, this is the food then that the choanoflagellates ingest. So they generate the water flow then with the beating flagella. They are heterotrophic because they rely on these organic particles as a source of food, and they're either single-celled or colonial. So making the jump now from the choanoflagellates to the animals proper, let's consider more of the characteristics of an animal and consequences of multicellularity. So instead of a colonial creature where you just happen to have a couple of identical cells that are still attached to each other, in true multicellularity, you have a much different kind of mechanical connection between adjacent cells. It should make intuitive sense to you that if you have an animal's body with multiple cells, such as shown here in this sponge, for example, that the cells will always be, that it would be difficult for the animal to remain viable if the cells became ripped apart from each other. And so it's very important that the cells can remain mechanically attached to each other. These kind of mechanical attachments are generated by an extracellular matrix. And so an important component of the extracellular matrix, which is unique to animals, are proteins called collagen. So the bit that's unique is the collagen. The extracellular matrix is not unique. So for example, cell walls are an example of an extracellular matrix. So it's not the existence of an extracellular matrix, meaning something outside the cells, which is an important structure. That's not the part that's unique to animals. The part that's unique to animals is the collagen. And I was looking for a nice picture to show you so that you have a better feel for what that looks like. And there is actually a nice picture from earlier in your textbook that, from chapter 11. And let's see. If I touch this, it's going to make it a little darker so you can see this better. So when you go through the Mastering Biology interface, you do have access to the entire electronic text. So you can access the chapter 11 if you want to read the accompanying text that goes with this. But anyway, just to orient you, this is a picture of the extracellular matrix in animal cells. And to orient you, here's the plasma membrane. So that's the outside of the cell, that's the boundary of the cell, right? This is inside the cell in that direction. So the cytoplasm is to the left. And this is then outside the cell, or the extracellular area, to the right. And so this is the extracellular matrix out here. This pink object here is supposed to be an example of a collagen fibril. And you can see that the different polymers that make up the extracellular matrix are intertwined and interconnected, even with the cytoskeleton of the cell through proteins in the membrane as shown here. So this then is really important for structural integrity. If one cell is pulling on another, then you don't have the cells being ripped apart, but they were remaining attached together. So this is a really important mechanical feature in multicellularity. So in addition to cells being mechanically supported and attached to each other with these extracellular matrices that have collagen in them, a couple of other things that are unique to animals are the existence of so-called nervous tissue and muscle tissue. So nervous tissue is made of nerve cells and muscle tissue is made of muscle cells. And a tissue, you may remember from earlier biology classes, is a group of cells with a common structure and function. So this is something one gets very commonly in animals. It is unique to animals, but it is not a synapomorphy for all animals because, in fact, sponges are an example of an animal that does not have nervous tissue, nor does it have muscle tissue. So unique to animals, but not found in all animals. For example, sponges. Sponges do not have proper neurons or proper muscles. Okay, let's spend a little time talking about body plans of different animals. So what is meant by the term body plan? This refers to a set of traits or characteristics that are a very different kind of overall functional design for a specific group of animals. And so all animals are divided into 29 phyla. This is a taxonomic group or taxonomic level. Singular is phylum, plural is phyla. So there are 29 distinct different animal body plans that have been recognized for a very, very long period of time, and they are listed here in table 31, which is in front of you right now. It's a very long, skinny table. I chopped it into two so that you could see it a bit more easily. And so what's useful about this table is that it shows the major groupings, but also how the different phyla are arranged in these different groups. Now, in a traditional invertebrate zoology class, one would just march through all the phyla one by one and cover characteristics of all of them. We're not spending that much time on the animals as a whole, so we're just going to pull out a few representative phyla for special coverage and treatment, and those are the key lineages, if you want, for which you'll be responsible, but we certainly won't have time to go through all of them. So in this first group here, the only phylum that we'll cover are the sponges, phylum Porifera. We'll also then cover, when we get to the protostomes in this particular group, we'll cover the flatworms, Platyhelminthes, and we'll also cover the mollusks, phylum Mollusca. So this will be Wednesday. We'll cover these two phyla, and then we will cover some of the ecdysozoans. We'll cover the nematodes and also the arthropods. So this will be on Friday. And then the following Wednesday, because Monday is a holiday, we'll cover two of the deuterostome phyla, the chordates and echinoderms. Okay, so those are the examples of some of the phyla that we'll pull out. So we'll go through these groups, general characteristics, but then we'll pull out a couple of phyla and go through those more specifically. So, earlier we talked about the Cambrian explosion, the Cambrian period, the first period within our own eon, the Phanerozoic, and all 29 phyla have representatives that go that far back, about 500 million years or so. Here's a picture, by the way, on the right showing the location of some of the really important fossils from the Cambrian era, and fossils continue to be discovered and found in some of these deposits. It is important to note that as we go through these animals, over 99% of the species that have ever been on the planet are in fact already extinct. And so, for example, in the fossils we find many rather extraordinary looking animals. This is one artist's rendition from the evidence of some fossils of what some of the organisms might have looked like back in the Cambrian that are no longer on the planet. So we're not going to spend any time talking about fossil forms. We'll just go ahead and talk about animals that are still on the planet, But the fossil record is a really important source of information for understanding the evolution of these different animal groups. But I think it's also significant to note that all of these 29 phyla do go back as far as the Cambrian. Okay, so as mentioned, the body plan is a set of traits or characteristics. One can use a lot of different items in the list. The traditional ones that have been used to characterize different body plans that are also used in your textbook are issues such as overall body symmetry, cephalization, germ layers, body cavities, the fate of the blastopore, cleavage type. These are all characteristics of an embryo, characteristics early in the development of all animals. And so we'll spend now a little time talking about what happens as a very early development in animals. Figure 3312 from your textbook, which is in front of you right now, starts with gastrulation. This is a stage in animal development. But what I think is tricky is to jump right on and start at this point. I think it's a little confusing for students because it's hard to put this in context, and one could reasonably ask, well, what happens before we get to this point of gastrulation? So we're going to quickly rewind, go back to the zygote, and then cover just a couple of major steps so that gastrulation makes a little bit more sense. So what happens during early embryonic development in animals? And this is most. There are variations on a theme. There are some animals that don't do this, but this is overall generally the pattern that one sees. I can go through this fairly quickly, first as text, and then as a diagrammatic representation. So most animals have sexual reproduction. Most animals are diploid in their dominant stage. They become diploid by having a haploid sperm fertilize a haploid egg. This forms a diploid zygote. That's the single-celled stage. It then undergoes cell division. The cell division, the very first cell divisions have a special name. They're referred to as cleavage. And what this means specifically is cell division without growth of the embryo as a whole. So that would be an example of that would be a cell divides into two, and each of those, if each of those cells is only half as large as the original cell, so you're no more massive than you were prior to division. So the zygote undergoes repeated cell divisions then without actually getting larger at first. Then there's formation of a specific stage called the blastula, and then this blastula undergoes a process called gastrulation, which was shown in that figure we were just looking at. And at this point then, the name of the embryo changes from a blastula to a gastrula, and this is a significant point in development because this is when the different embryonic tissues are laid down. And then a number of different developmental genes come into play, one set of which are the so-called Hox genes, and these control anterior-posterior development, and we'll talk about those a little bit more later. So that was text. Now let's do a diagrammatic representation of exactly the same information. So, starting up at the top left, here's a zygote. Single cell diploid. It divides, it divides, it divides. Of course, when it first divides, it goes from one cell to two to 4, 4 to 8. At this point here, we have an 8-celled stage, so we've had a number of different divisions that are sort of glossed over. Notice that this 8-celled stage is about the same size as the zygote, which is what's meant by cleavage not getting any larger. You have a larger number of smaller cells. They divide and they divide and they divide and they divide. Eventually, one ends up with a different stage. This is now the stage called the blastula. It looks like a little ball. If you peer inside, as shown here, you can see that it's actually a hollow ball. You can't tell that from the outside because it's a completely enclosed sphere of cells. Then what happens is at one end of the blastula, there is an invagination. This process is called gastrulation, where this new structure forms. And now at this point, this embryo is now referred to as a gastrula. Okay. Let's watch this now in a movie form. It'll make more sense, and I'll dim the lights a little bit so you can see what's going on. So up here we have a time-lapse movie. These are actually eggs. They've just been fertilized. You can even see the sperm on the outside. You can tell they've been fertilized because of the structure that formed on the outside there. There we have one cell forming now two, dividing into two. So cleavage has started. Now the two are going to divide into four. Okay, so now we have 4 cells, they'll divide into 8, so on, and we just jumped over a few cell divisions here. We're now at the stage of a blastula, and it's somewhat transparent, which is why you can see through to the inside. So this blastula is now beginning gastrulation. Here we have some of the cells moving. So here's the formation of this invagination here. So this is now a gastrula. We now have this structure forming right here. Okay, so this is going through all the way to the other end, but the movie stopped before that occurred. Okay, so now we're ready to jump to figure 3312, because now we know where our blastula, our gastrula came from, right? So diagrammatically up here at the top of figure 3312 is a picture of the gastrula with gastrulation going on here. Again, there's a still from the movie to remind you what the real thing looks like. And in this case, then, if it continued, rather than stopping it in time, if we allowed this process to continue, then what would happen, I suppose I should get a yellow marker here. Over time, then, this would go all the way through in time, and eventually one would end up with a complete tube. Similarly, over time, this would continue extending and then eventually break through the other side, and one would end up with a tube. One would end up with this basic pattern then for the example of animals with so-called complete digestive systems, meaning their digestive system has 2 openings. It goes mouth to anus. Now there are 2 primary groups of animals that form in this way. They're called protostomes and deuterostomes, and the major difference is just simply whether or not that initial point of invagination, where the blastopore is, whether that becomes the mouth of the overall complete digestive tube, or if it becomes the anus of the overall digestive tube. So these were characteristics that were identified many decades ago by the earliest zoologists that studied animal development. It was recognized that there were 2 primary different groups of animals that had either the feature shown on the left or the feature shown on the right. This was prior to the ability to use molecular characters. And now that we do have the ability to use molecular characters to substantiate phylogenies, they have verified that in fact protostomes and deuterostomes really are separate lineages. And so the names come from these earlier embryological observations, although if one looks at the embryo after the complete digestive system and the embryo has formed, you can't really tell looking at it which way it did in fact form. If one takes the embryo then and chops it in a plane like this and then looks at it sideways, then one gets a view which is shown here down in the bottom, right? So now we're looking at the digestive tube end on. It's going, that's the yellow structure right here, labeled gut, right? That's the digestive tube, so it's coming in and out of the board at you. And this shows the 3 different so-called germ layers. These are the important embryological layers. So there's ectoderm on the outside. You can probably guess that from the prefix ecto, and that is shown in blue. We have mesoderm here, and then we have endoderm, which you can't see because yellow is too hard to see, so I'll go ahead and write that in another color as well. So these are the 3 different embryologically derived layers that give rise to all the tissues of the body that are found in animals that do have 3 layers. Not all animals do have 3 layers, but if they do, these are the 3 that they have. One can follow the fate of these different early patches of, or these early germ layers later on in the adult form of the body, and that's shown here in figure 3310, which is now in front of you, which shows cross-sections of a number of different kinds of worms, which are very different in structure, although from the outside they all look, they're all worms. We'll spend more time talking about worms in the next few days. And if one then looks at the body in cross-section, one always sees the ectoderm on the outside, then the mesoderm, and then the endoderm and the innermost part of the animal. And so this is described as a basic structure of tube within a tube, or you could think of it as a tube within a tube within a tube if you include all 3 layers there. And the ectoderm typically is what develops into skin and also the nervous system. The mesoderm develops into the muscles and some of the other viscera, and the digestive system develops from the endoderm. And that's pretty universal across all animals that do in fact have these 3 layers. The other thing that I want to point out to you is that in this structure there is, in this diagram, there are different kinds of body cavities that are pointed out here. So one cavity that is diagnostic of certain lineages of animals is a so-called coelom, and this is a body cavity that can be recognized because it occurs actually inside the mesoderm, as shown here. So you can tell in this particular diagram because it's surrounded by a red color of tissue, because red is used to identify mesoderm in this particular tissue. Not all organisms have large or really obvious coeloms, and so the functional significance of coeloms are not that different from other kinds of cavities, such as the pseudocoelom shown here in this other worm, which is called a pseudocoelom instead of a proper, regular, true coelom, because it's not completely surrounded by mesoderm. So it has arisen in a different way embryologically, but it is still a fluid-filled cavity, and so it still has many of the properties and features of a true coelom. Over the quarter, you have seen a number of different kinds of life cycles. You've seen protist life cycles and plant life cycles. And now we're going to look at a much more familiar cycle, and that is an animal life cycle. Very simple and straightforward. Figure 3314 in front of you right now, which features a sea urchin as the representative animal for this life cycle. So this is going to seem very easy and familiar compared to some of the life cycles we have looked at. So starting down here at the bottom, we have a diploid organism which undergoes gametogenesis, or formation of gametes, which are haploid. We've got fertilization leading to a zygote, which then undergoes cell division to form a multicellular diploid individual. This should all be completely familiar. The only things that might be a little unfamiliar to you are some terms. Larva and juvenile are actually technically meaningful terms that you might be less familiar with in terms of their exact meaning and usage. The term larva is specifically used to describe an immature stage of an animal that does not look like just simply a miniature of the adult form. So in the sea urchin, for example, you can tell that this larva really does not look like a little miniature adult, and so it is described with the term larva as the proper way of describing that immature stage. Juvenile is a term that is used for an immature individual that looks like basically a little miniature replica of the adult form. So you can see that by this point in the life cycle of this particular sea urchin, it still was not an adult sea urchin, but now at least it looks like a little tiny miniature adult sea urchin, and so that's why it's called a juvenile at this point. The term metamorphosis is used to describe a change in form from a larva to a form that now looks more like the adult. So metamorphosis doesn't have to be to a juvenile stage. It can even be to an adult stage, but it refers to a large change in form. This is very common in animals. Here's another example of an animal that has a larval form. Mosquitoes. There's a mosquito adult in the bottom. So a mosquito adult flies around in the air. It has wings. It sucks blood if it's a female. And mosquito larvae, completely different. They live in water, underwater. This is a picture taken underwater of some individual mosquito larvae. Completely different in appearance, structure, habits, what they eat, how they live, and so on. So very, very different. So the term larva is used here because to indicate they're completely distinct and different from the adult stage. Okay, we're now looking at figure 33.2 from your textbook in front of you right now, which is the phylogenetic representation of the major animal groups. Now with some representative phyla shown in here. So we already considered multicellularity as a synapomorphy for animals as a whole. Again, notice up at the top here we have choanoflagellates as the outgroup for all of the animals. And I want to just point out a couple of different things here. Let's get that out of the way. These terms here, diploblasty and triploblasty, refers to how many germ layers are there. Triploblasty means that there are 3 germ layers, so these are the familiar ecto, meso, and endoderm, the 3 different germ layers that we have just been talking about. The diploblasty is just simply refers to having 2 germ layers. So this is ectoderm and endoderm without mesoderm. So these animals tend to be much, much simpler in structure. And you can see that there's a relatively small number of representative animal phyla that only have 2 germ layers. So by far, most animals have 3 germ layers. Let's see. The other thing I wanted to point out here are some of these other names. Bilateria refers to those animals that exhibit so-called bilateral symmetry in their bodies. And these are all divided into 2 groups, each of which are monophyletic, the protostomes and the deuterostomes. So let's consider symmetry then. This is not a figure from your book, but I thought this might help make symmetry a little bit clearer. Body symmetry of animals is usually described as either being radial or bilateral, as just a simple dichotomy here. So we've got an example of radial up at the top and bilateral symmetry on the bottom. So what radial symmetry refers to is a general body form where one can take a plane and divide an animal in 2, and each of those halves is a mirror reflection of the other, and then one can rotate that plane around the center of the axis, and there are planes of symmetry as one rotates the plane. So one could also have it here and there, right? So does that make sense? This is referred to as radial symmetry because it's the same all the way around. So an example of a familiar object that also would exhibit radial symmetry would be something like a flower pot, where you can put a plane of symmetry anywhere down the middle, and each of the 2 halves would be a reflection of the other half. This is in contrast with so-called bilateral symmetry, in which one has a left and a right. So we're bilaterally symmetrical organisms. For example, and our plane of symmetry is right down the middle of our bodies. Our left is approximately a mirror image of the right. So notice that there is not another plane of symmetry. If you take that plane, cutting this lobster into a left and a right, and you rotate it around the axis, you do not get a plane of symmetry if you rotate it into this way, right? Because now you have a back that is different from a stomach. You have a dorsal surface which is different from, which is the back, from the ventral surface, which is the other side, right? So you have differentiation left, right, front, back in an organism, as well as 2 ends. So a familiar example of an object that would have bilateral symmetry would be something like a shovel. Now, don't get too wrapped up in hair splitting about symmetry. What do I mean by that? So if we go back to this example, is it true that every single organism in this group is every single one of these individuals perfectly bilaterally symmetrical? Well, no, that's not true. So for example, if you consider some kinds of worms and you look really carefully at them, you might think, well, sheesh, I look at this worm and I can't really tell the dorsal surface from the ventral surface. It kind of looks radially symmetric to me. I would absolutely not argue with that. So this is not, this is a general description about body form for this group as a whole. It absolutely does not unambiguously identify every single organism in this whole group. Similarly, some of the organisms that are not bilaterally symmetrical, if you really look at them closely, there are differences in parts of their body that make them look a little bilaterally symmetrical. And of course, we're not perfectly bilaterally symmetrical, right? Our heart is slightly more to the left side and so on. Our appendix is only on one side. So don't take this as perfect reflection or a geometrical imperative at all. It's a description. More significantly, most of the bilaterally symmetrical organisms have something, are associated with a process called cephalization, which is a concentration of sensory organs and also neurons at the head end. And of course, if you have a concentration of neurons at the head end, we refer to this as a brain, right? So if one looks back at the bilateral organisms, then to a certain extent, all of these organisms also have some degree of cephalization. You can see this is identified as a synapomorphy here. So there is only a single organism which is described as being having bilateral symmetry without cephalization. So this is a really important association. And this has significance. Let's see if you can figure out the functional significance of this. So considering an animal that has got radial symmetry, and this is an any answer counts clicker question, would you expect that, what would you expect the functional significance to be? What would an animal with radial symmetry possibly be able to do better than one with bilateral symmetry? A, move quickly in one direction. B, direct threats from above or below equally well. C, deal effectively with food distributed homogeneously in 360 degrees. Or D, focus attention in a single direction. Okay, this is an example of a typical kind of test question one might expect about the significance of radial versus bilateral symmetry. I'll give you 20 seconds or 30 seconds or so to discuss it with your neighbor, and I'll see what you guys think. Okay, everybody got a chance to think about this and click in? All right, looks like it's stabilized here. Okay, so here are your answers. Well, excellent. Yeah, this would be considered, in fact, the typical argument that's made for under what conditions would radial symmetry be advantageous. Nice job. I'll see you guys on Wednesday, and we'll start with sponges.